and then we'll finish that on Thursday, and then finally on Friday finish up with acid base balance. But remember we had 2 locations for the 2 types of kidneys by location. We had cortical nephron, which the nephron loop did not dip into the medulla or only dipped into it partially. And then we had the nephron whose capsule was very close to the beginning of the medulla and its loop dipped down far, and that was our juxtamedullary. Okay? However, for both types of nephrons, remember the structures that we found in the cortex remain the same. So we have the capsule, and we have the proximal and distal convoluted tubules. So that function of filtrate formation remains the same for both types of nephrons. As we'll see on Thursday, the function of the nephron loop is important only in the juxtamedullary nephron. Okay, we'll look at that function on Thursday. So let's go back and look at, well not go back, but if you'll look on page 20 of your lecture notes, you'll see a diagram that looks like this. Not page 20, sorry. Turn my pages back. Page 12. You'll see a diagram that looks like this, and it shows the intimate relationship between the blood supply of the kidney and the nephron structure. And that both of those are required for filtrate formation and modification. So over on the upper right, I have 3 components listed that are involved in urine formation. Now urine is what we call the final product. However, remember at what point, what structure does this fluid enter before we actually call it urine? Minor calyx. So while it is called filtrate, as soon as the fluid leaves the blood and enters the nephron, and until it leaves the collecting duct, so from nephron it goes to collecting duct, until it leaves the collecting duct and enters the minor calyx, that entire fluid is filtrate.
Are there notes not gone?
There are. And so these are the 3 steps that we're going to start looking at today. So if we look at the origin of our basic nephron, starts out as the glomerular capsule. And within that capsule, we have our peritubular capillary. And so the process by which material is moved out of the capillary into the capsular space is known as filtration. And this is separation based on size, and as we'll also see, the presence of charge. That creates our fluid, which from here until we get to the minor calyx is known as a filtrate. I'm going to modify this slightly from the diagram that you see there. So this is our proximal convoluted tubule. Remember, isn't it small enough and doesn't have too much of a negative or positive charge, is able to pass into the filtrate. And that's way too much that we want to get rid of. All right, so if we were to just look at filtration, your body would make about 180 liters of filtrate every day. That's way more blood than you even have in your body. So obviously, we're not sitting on toilets with hooked up to IVs eating ice cream sandwiches. So we reabsorb most of that back into the body. That process takes place in the proximal convoluted tubule. So our afferent arteriole becomes the peritubular capillaries. start moving material from the filtrate back into the blood. And that process is known as that would be the efferent arterial giving rise. Did I say afferent? Sorry. That would be the efferent arterial giving rise to the peritubular capillaries. So moving material from filtrate to blood is reabsorption. We actually take some of the material that wasn't secreted and move it from the blood into the filtrate. So going from blood to filtrate, increasing from the amount that was actually filtered and adding to it even more is secretion. So we filter sodium, and we'll put this up on the board. Sodium, glucose, chloride ions, amino acids. We reabsorb 80% of the sodium and all of the glucose and all the amino acids at the proximal convoluted tubule. And I'll be writing that a little bit later. We're going to add hydrogen ions and some more urea and the breakdown products of antibiotics. So we're going to have even more of that in the filtrate than we actually had in blood. Now we form our loop. So this is the nephron loop. This process is what we're going to cover on Thursday. And what we have happening here is primarily reabsorption. And the purpose of the nephron loop is to form a concentration gradient. That's half of Thursday's lecture, so I'm not going to say any more about it than that. Okay? It does not concentrate the urine, but actually the filtrate, but makes a concentration gradient. Our filtrate is actually less concentrated when it leaves than when it comes in. Then we form our distal convoluted tubule. And again, we do secretion, reabsorption, and secretion. But this is typically regulated, right? So this is typically regulated under the influence of hormones. That's our nephron. End with the distal convoluted tubule.
So on the nephron loop, then there is no secretion then?
Not typically. All right, our distal convoluted tubule then joins the collecting duct.
Oh man, I have no room.
And now as the filtrate flows down the collecting duct, this is where concentration occurs. and by concentration, we're going to reabsorb sodium and water. Again, under hormonal influence. And that's our lecture for the next 2 days. Today we're going to focus on this area. So the capsule and the proximal convoluted tubule.
Can you get a picture? Yeah, thanks.
So why do you draw that line?
That's the separation between the cortex and the medulla. Remember I said the nephron loops of the cortical nephrons that stay in the cortex aren't very important. It's the nephron loops that dip down into the medulla that become important. So now let's start with that first step, filtration. And that takes place across our filtration membrane. So with your permission, if you're done with the material on the board, I'll erase that. And then I'll diagram our capsule and our filtration membrane. Remember the respiratory membrane? That was a sandwich of two simple squamous epithelia, the basement membrane between the two, maybe a little bit of connective tissue. And our filtration membrane is also a simple squamous sandwich, and just the basement membrane between. So may I erase the board? That's okay with that? So when we look at the capsule, the word renal corpuscle up there, I have it up on the screen. It's used in textbooks. I don't really like it because corpuscle means cell. This obviously is more than one cell. But the term corpuscle refers to both the capsule and the contained blood vessels. So it's a term for the combined component that both are required for filtration. All right. So I'm going to draw this capsule way over here on this side, and the outer layer of the capsule, the outer, the parietal layer, is formed of just regular, simple squamous epithelia. And remember, you could either call it the Bowman's capsule or the glomerular capsule. Either term is fine. Parietal?
There's a T to right.
I leave out a T. And then coming into that capsule, remember we have our afferent arteriole. And it forms a ball of capillaries. And then leaves as a more narrow efferent arteriole.
I'm so sorry.
Intimately associated and wrapping around the epithelia of the capillary bed is our visceral layer. And so when we look at this in the microscope slide, it's very difficult to tell which nucleus belongs to capillary and which nucleus belongs to the capsule layer. So the blue is going to be the visceral layer of the capsule. Suppose I can write it in uppercase. It also is simple squamous epithelium. And it is unique because it has these extended legs or foot-like processes. And so these cells are actually known as photocytes. Yes.
So that's the visceral layer of the capsule?
Uh-huh. So the parietal layer of the glomerular capsule, or Bowman's capsule, and the visceral layer of the capsule.
Thanks.
Damn, my drawings are getting good.
And so this would be considered to be the capsular space. And filtration is going to take place across that layer. And so in the capsular space, we find our filtrate. As the filtrate is formed, then it flows out into our proximal convoluted tubule. So with our capsule, we have one opening, kind of, that allows the blood vessels to enter in. And then we have another opening where the filtrate is leaving. So this area is considered to be the vascular pole, where the blood vessels are coming into the capsule, kind of like the hilus, but we don't use that term. And then this area, even though it's filtrate, is identified as the urinary pole.
It's PCT. Oh, proximal convoluted tubule.
So again, on the diagram,
Proximal
this would be the
convoluted
vascular
tubule.
pole, this would be the urinary pole.
So this is the first place in the capsular space where we find filtration?
Yes, this is the formation of the filtrate.
You're talking about where the, right where it leaves
Right?
capsule.
So
Comes in.
where the blood vessels come in is vascular, and at the other end, where the filtrate is moving into the proximal convoluted tubule, is urinary.
You're talking about the ball part.
Right.
Is that part, not the tube?
So if we look at a model,
It's like north and south pole, called vascular pole, and the
so here's what I've diagrammed. Okay, so this would be the vascular pole of the capsule, and this would be the urinary pole of the capsule. Okay, so just talk about that part there. Okay, so now let's look at the relationship of our actual filtration membrane. Okay, so we're going to take these two cells. The blue is the visceral layer of the epithelial capsule, and the red is the simple squamous of the capillary. And let's look at that in a little more detail. Here's a, this would be the vascular pole here. You're not seeing the blood vessels quite as easily. And this one's not showing the urinary pole, but sometimes you can get a cut where you can see it right down the center the way it's seen here. So here is a light microscope image. This would be both the photocytes as well as the capillary epithelium. It's hard to distinguish between them. Here would be our capsular space, and here would be the parietal layer of the capsule. Very common image in Bio430 for looking at simple squamous epithelia. Okay. Now this is a scanning electron micrograph of just the glomeruli. So we have the afferent arterial is glomerulus, and then the smaller efferent arterial levy. And the nephron structure is removed. So now let's look at the relationship between those two. So one of these fluids is plasma, and one is filtrate.
That was a clear.
So the two epithelia forming the sandwich, the bread of the sandwich is here and here. The basement membrane, so here would be the light and half the dark, that would be the basement membrane of this cell, that would be the basement membrane of this cell. So which one is most likely, do you think, to be the endothelial, the capillary membrane? You're right. So look at the unique arrangement of those. You're actually looking at two cells on that upper layer. This is the appearance of a scanning electron micrograph of the podocytes. And remember, podocytes is our term for the visceral epithelia. So we can't see the capillary, it's inside of this. And these are like, almost look like tree roots or an ivy that's grown prolifically around and wrapped around that blood vessel. So the nucleus of the podocyte would be right here. It sends out these branches, and each branch has foot-like processes, they almost more like finger-like processes, and those interdigitate with another cell, creating a narrow space between the two called a filtration slit. So let me draw that component first. So in this upper region here, I'll leave this diagram up here and just draw it over here. So this upper space up here is going to be the capsular space containing filtrate. The space down here is going to be the glomerular capillary. And it contains blood.
Capillary?
Okay. And I'm going to diagram the filtration membrane in between. So forming the visceral layer of our capsule, we have our podocyte. And it has these foot-like processes that extend down and rest on the basement membrane. It's a simple squamous epithelium, right? And these foot-like processes are identified as a pedicel. I'll draw another one, purple this time, so you can tell the difference between. And its foot-like processes are going to come in between So this would be the visceral epithelium, and the gap between each of these foot-like processes is called a filtration slit. So if I make that a little bit larger, that gap filtration slit, and there's actually proteins that span that gap and create a filtration slit diaphragm. Now, this epithelium has a basement membrane, okay? So there's going to be the epithelial component of the basement membrane and connective tissue component of the basement membrane. So I'm not going to require you to know the terms, but in electron micrograph, they're called the lamina rara and the lamina densa, the light and the dark areas. So I'm just going to put I could put the in, but you guys would have a different connotation, and I don't want to go in that area. So that's one layer and part of the filling of our filtration membrane. The next layer is the epithelial layer of our capillary, the endothelial cell. And these are fenestrated capillaries. Remember the term fenestrated capillaries? Those have pores in them. All right, so
Oh, do you need green?
Awesome, thanks.
So this layer also has a basement membrane. That's the filling of our sandwich. And these would be the fenestra or pore. That's the filtration membrane. Okay? So if we look at another electron micrograph, this is what I've drawn on the board. So these two pedicels here would belong to one cell. These two pedicels here would belong to the alternating interdigitating cell. This would be where filtration is located. The light and half the dark would be its basement membrane. Here's the epithelium of the capillary. Here would be the fenestra. And here would be its light and its dark component of its basement membrane.
Could you remind us the holes, the pores are in the membrane or in the capillary?
It's a continuous basement membrane. So the pores are the capillaries. It's like you were to take an air mattress, you're floating out on the tube, and take a really large hole punch, and punch a hole through the apical and basal membrane, but when you punch that hole, it's sealed. So if you look at the surface of this cell, and I'll erase this in a moment. So if we look at the surface of this cell, here's our epithelial cell. It has pores passing all the way through it. A channel. So punch the hole, and it's sealed all the way around it. So fluid particles can pass through that cell without ever coming in contact with the cytoplasm.
But the basement membrane itself is.
The basement membrane is continuous.
Okay, thank you.
So now if we look at the diameter, the pores have a much larger diameter than the filtration slits do. Okay? So the three factors that are really effective in the filtration are, one, the pedicels have, do you guys remember what a glycocalyx is? The coating, the cell coat, is the glycoproteins and glycolipids extending out of the lipid membrane. It makes it look fuzzy when you look at it in electron micrograph. Okay, that glycocalyx of our pedicels has an overall negative charge. So that's one factor that prevents molecules from entering into the filtrate, even if they're small enough to cross the plasma membrane. So one factor is the negative charge. on the glycocalyx of the pedicels. The other factor is the narrow membrane of the diaphragm. So the filtration slit and its diaphragm. So the small diameter. Of the filtration slit and its supporting proteins. And then the third is not the fenestra, but the negative charges found in the basement membrane. Yes?
And what are all these three things then?
These are the three major factors of what gets out of the blood and into the filtrate. So factors determining the filtration parameters. So our openings that are present in the fenestra certainly keep the blood cells out, they keep large proteins out, but they're not small enough to keep proteins like albumin out. But we don't have albumin in our filtrate. So what keeps the smaller proteins that would be able to pass through the fenestra are the negative charges of the basement membrane, pedicels, and the more narrow openings of the filtration slits.
Is it negative charge for the proteins?
Yeah.